Welcome back, everybody. We just finished talking about just the structure of DNA, and that is going to be a huge help just understanding that structure and how DNA is then replicated, right? Watson and Crick theorized, like, we now understand how DNA is replicated and how um, inheritance occurs, right? It's because of that specific pairing. So now let's get into the details of how an entire strand of DNA is copied, right? Copying that entire strand is called DNA replication. So now let's go back into into these slides and look at those details. Okay. All right, so we talked about that history and the explanation of the structure. Now we're going to talk about replication. So here we've got another piece of DNA. And as I said, there's this 3' end and this 5' end. And that's really just telling you where, which direction the sugars are going in and the phosphate coming off that sugar. So that's the backbone. And A is always paired with T and G is always paired with C. So now the replication of DNA is called semi-conservative. Why is it called that? That is because um, if we think of this original strand here, and I'm going to make I figured out how to make this a little skinnier. Okay, this original strand, it's got these 2 strands of DNA, right? You've got this one on the left and this one on the right, and these are gray. If we want to make a copy of them, well, we're going to have to split those 2 strands apart from each other, okay? It doesn't get copied as both of them, like there's nothing that's copying both of them off of each other. So they get split apart, right? One goes to the left, one goes to the right. And then the left one gets a copy and the right one gets a copy. So if you know this left one is A, T, G, C, and therefore the right one had been T, A, C, G, right? You can then immediately figure out what the other one is. And so a copy is made from those 2 originals. And so in the 2 new strands that we have, in these 2 new strands, you have the remains, you have one old strand, which is gray, right? Those gray ones. And then you also get a new strand from that that they're showing you here in blue, right? So these are your 2 new strands off of that. So you've conserved the old and you've made new ones. So each new strand of DNA has one old strand and one new, so it's semi-conservative. You're keeping half of the old stuff. Okay, so what are the steps of replication? As I said, the first thing you're going to need to do is separate those 2 strands. You can't copy one strand if it's attached to the other one, right? So they get unwound and unzipped. And there's an enzyme that comes in called DNA helicase, and it is the unzipper enzyme. Okay, so this unzips them. And where it unzips them is called the origin. Okay, so if you've got this long strand of DNA and it unzips it here, right? You've got these 2 strands and they get unzipped. Right here where it starts unzipping is our origin. This unzipped part, if we consider this a zipper and you're going along and then it's like a fork in the road, right? It goes in 2 different, it splits. You've got one strand going one way, the other strand going the other way, so that's your fork. That's your replication fork. Okay, so how is the DNA then, how is it copied? Okay, so we've now split it up and now we need to make a copy of each strand. So each strand gets its complementary strand built off of it based on what is read on that strand. That reconstruction always moves in just one direction. So we talked about that 5' and that 3'. That's the direction that the new nucleotide bases are put down on. And I'll show you that this ends up making things a little bit, it's not the most efficient system, but it is a system that evolved, that things always get put down in one direction. And how does it get put down? Well, there is another enzyme called primase that figures out, okay, you've got an A here, I need a T, and it grabs a T. You've got a G, I need a C, right? It finds what it needs. And then there's an enzyme called polymerase that's going to actually attach them. So this is like a whole assembly line process of making the complementary strand of DNA. Okay. So here we've got our double-stranded piece of DNA. It's been split here at this origin, right? And you can see these replication forks, right, where it starts to split. Here's a fork, right, and here's a fork. Okay, so this is getting copied from the 3' to the 5', the 5' to the 3'. But that means it's 5' to 3' on the new strand, okay? So the new strand, if we're looking at this top one, it gets copied from, this is the 5 of the new one, and it carries along, right? And as it moves this way, this is going, these two strands of DNA are going to get continually further and further separated, moving down. So this blue triangle here, they've labeled it. This is helicase, okay? So the helicase is going to move along, just unzipping as it goes. And so as it's unzipping, this strand of DNA can just keep making a new one, okay? So it's just going to kind of move along that path. The helicase is unzipping, and the primase and polymerase are putting down the matching nucleotide bases in this 5' to 3' direction, okay? But then if we look on the other side of this origin fork, to the right side of the top strand, this is getting pulled apart as well by the helicase. It's unzipping. So we've got two different helicases unzipping in two different directions, right? 5' to 3' is still moving from right to left for the top strand, but it's unzipping from left to right. So your strand is getting built from right to left, but unzipping from left to right. So what does that mean? Well, when it's unzipped only this far, a piece of DNA is made this much. And then it unzips a little further, and it can make another little piece of DNA. And then it's unzipped a little further, and it makes another piece of DNA. So while we get one long strand on the left side of this fork, we're getting these short little strands on the right side. And on the bottom strand of DNA, down here, it's the opposite. So this one from 5' to 3' can move from left to right. And that's the direction that this DNA helicase is opening things on the right side here. So this, we've got one long leading strand that just keeps getting replicated. But to the left of the origin here, you know, it's just opening up a little bit, it makes this piece of DNA. It opens up a little bit more, it makes this piece of DNA, right? So we get these little fragments. These little fragments are called Okazaki fragments, named after the person that figured this out. So we've got these tiny little fragments, and they're going to have to get all joined together at the end. So I have a video to show you that explains this process really well. So let me get to that. Okay, and let's go full screen here. All right, so you've got this video.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase is called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up fragments of DNA in both strands to form a continuous double strand. DNA replication is semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, so let me get back to our slides here, but I think she does a great job of explaining it with the video. Sometimes you just have to see it happening to understand it. Okay, so let's get back to where we were. Okay, so that is how DNA is copied. Okay, you now understand the steps of replication. First, you need to separate those strands with helicase, and then once those are separated, primase can put down a little starting primer, and then polymerase fills it in, right? So that is how an entire genome is copied. And this is very useful for mitosis, right? We need the entire genome copied in order for that cell to replicate. So this is for mitosis. But you don't always need the whole genome replicated. Sometimes you just want a little section replicated, just a gene. So this is for transcription, translation, and gene regulation. This is a different process. And we can understand this by looking at this little picture here. So what is your DNA for, right? It's not just so that you can copy it from one cell to another. Ultimately, it must have some purpose, right? And the purpose of DNA is to ultimately code for a protein. So the DNA is in your nucleus. Little sections of it get copied into a messenger RNA, and that messenger RNA gets turned into a protein, and that protein is used by the cells to do something, right? That lots of proteins do lots of different things, but that is the process here. This is our flow chart. It goes from DNA to RNA to a protein. Okay, and so in the next lecture, we will explain how all of that works.